Howdy, folks. Thanks for checking in to Mr. Ulrich's Land of Biology. I am Mr. Ulrich. In this Notescast, we're going to be taking our second look at Gregor Mendel. If you remember in the first video, we looked at how Gregor Mendel did his studies and the laws of dominance, as well as how to do a Punnett square. In this video, we're going to be looking at the other two laws of inheritance, looking at the law of independent assortment and segregation, as well as looking at dihybrid crosses. So I'm just going to go quick change my shirt. Let's take some time to talk about Mendel's laws in a little more detail. His first law has to do with segregation. Now, we talked about meiosis last couple videos. Take a look at those if you need to. But we know that homologous pairs of chromosomes segregate during meiosis. They're going to bring their alleles with them. So that means that each gamete is going to get one of each homologous pairs of chromosomes. Now, when does all of this segregation take place in meiosis? Of course, during metaphase 1, because that's when the tetrads actually line up. We also get the crossing over, but that's metaphase 1. Following one trait at a time is what we call a monohybrid cross. And Mendel did this with flower color and seed color and all those other several different characteristics that we talked about. Mendel also followed the inheritance of two characteristics at the same time. So looking at both seed color and seed shape, or pod color and flower color at the same time. For example, here's the dihybrid cross between yellow round peas. So we're looking at pea color and pea shape versus green and wrinkled peas. Again, same pea color and pea shape, just different pea color and pea shape. And you notice that in the F1 generation, all of the offspring were round and yellow. So meaning that now we know that that one parent generation, one of them was homozygous dominant for both pea shape and pea color, and the other one was homozygous recessive for both pea shape and pea color. In the F1 generation, all of them are going to be heterozygous for both traits. When we did the self-pollination, you get this kind of interesting and characteristic ratio of 9, 3, 3 to 1. What Mendel was trying to figure out with these dihybrid crosses is if genes, or in his case factors, since the term gene hadn't been really coined yet, if these factors, when they're inherited, come in units. Do they come together, or do they actually segregate independently from one another? What this means is if you get a recessive allele from for one trait from that parent, do you get other recessive alleles or other traits from that parent as well as kind of like a unit, or do they independently segregate so that you have all different combinations of dominant and recessive alleles for either trait? So let's check it out, which way actually works. If they're hooked together, when you get a dominant allele in a gamete, you're going to get the other dominant allele in that gamete. When you get the recessive allele in a gamete, you're going to get the other recessive allele as well in this case. So the Punnett square is very simple. It's going to look just like we did for the monohybrid cross. And we'll get the same phenotypic ratio of 3 to 1. But what we actually see here from the results is a 9-3-3-1 ratio. So clearly this is not working. So let's look at the other way. Now, this is going to be a little bit more complicated because there are more choices. There are more ways that those alleles can arrange. I can either get the dominant Y and the dominant R allele, or I can get the dominant Y and the recessive R allele, or the vice versa, the recessive Y and the dominant R. I can get both recessive Y and R. So I have each one of those different types of gametes that are possible. Remember, a Punnett square is to work out the possible gametic combinations. When we start putting these together and fill out the Punnett square with all the possible combinations, we can see here are the 9-3-3-1 that we're trying to get to match, I should say, from the experimental results. The results of these dihybrid crosses led Mendel to develop his second law of heredity, one of his shakier ones, called the law of independent assortment, which states that different factors, which we now know are different genes or alleles that are located at loci, separate into gametes independently of one another. So if I have two traits like yellowness and greenness versus roundness and wrinkledness, and I have an individual who is heterozygous, they're going to go through meiosis, and in one way they can make different gametes. And if the chromosomes line up a different way, then they can make a different array of gametes. And there's an equal chance of all of those different gametes being produced. Now, this does work for chromosomes, or for traits, I should say, genes that are on different chromosomes for sure. So if they're non-homologous, then they are certainly going to align independently, and they are certainly going to be segregated independently from one another. And this is going to produce a wide variety. But there's actually even more variety than that. What we've actually noticed is that they don't even need to be on the same chromosome in order to be inherited independently from one another. There are so many chiasma and so much crossing over that takes place that, especially when we start getting further and further away from the centromere, crossing over is so routine and so regular that they're going to act as if they segregate independently from one another, even though they're located on the same chromosome. As with the law of segregation, independent assortment is also taking place during metaphase 1. The big exception here are those traits that are located really close to one another and close to the centromere, so that they can kind of get around some of that crossing over that shuffles the rest of the genes that are further away. These tend to be inherited together, and we call them linked. So traits like red hair and freckles, things like this that tend to be inherited together, are linked. The key to Mendel's success as a researcher, and the key to most researchers' success, was meticulous and painstaking note-taking and data crunching and data analysis, all the way from gamete formation all the way up to the breeding individual. Mendel then was able to determine the laws of segregation by doing monohybrid crosses, looking at those single traits and determining that each characteristic is controlled by an allele that segregates into different gametes. So each parent donates one of a pair of alleles for a particular trait. Independent assortment, which he determined through the dihybrid or even more crosses, showed that the segregation of alleles takes place independently from one another. We know now, of course, that there are these linked traits that are on the same chromosome, close together, and closer to the centromere to avoid the shuffling of crossing over. Mendel's choice of pea plants as a test subject was a good one. There's lots of different traits that we can look at in pea plants that are obvious, not hard to see: flower color and pod shape. Plus, it's really easy to control which plants are mating with whom. We can bag plants, we can cut off anthers, we can paint pollen onto the female parts of other plants. Significant amounts of luck involved here. He also chose traits to follow that were basically a monotype that is controlled by a single pair of alleles. And he also chose traits that showed complete dominance and recessivity. As we're going to talk about in future videos, not all traits follow simple Mendelian genetics, but this was a fantastic start. And amazing that he was able to determine all of these things without knowing what DNA was, the importance of it, how it functioned, or any of the stuff that we now kind of take for granted. Oh, but we'll talk about non-Mendelian patterns of inheritance in the next video. For now, thanks for checking into Mr. Ulrich's landofbiology.com. Again, I am Mr. Ulrich, and if you have any questions, feel free to drop me an email, write them in the comment section below, any of those things. Other than that, we'll see you in class.